We'll start with GS Galaxy project update. Then we will get hands-on to run through the tools currently we have in the Galaxy pipeline and to receive your feedback. So I'm the breeding application specialist of GOBii project. So I lead this GS Galaxy pipeline development project team. Here we also have Venice at the back. She's one of our lead developer, writing different existing tools in Galaxy in the back end. Also, IRRI has been fundamental in starting this EIB demo server. So including the tools Milka has showed you this morning. So IRRI is the hub. It's very important centralizing all these EIB Galaxy tools developed by different projects, including HTPG, GOBii, or including the forward breeding. You can see we are still in the process consolidating different tools based on the functionality groups. So you will have a different look on the Galaxy pipeline. So here I'd like to give you just a few slides to oversee the project. Basically, the website here, what I want to tell you is you can go to gobiiproject.org. From there, go to product. Under product, you can find GOBii loader, extractor, QC, flapjack tools, and genomic selection Galaxy pipeline. We are in the process updating our user documentation. Also, we'll share a tutorial as a YouTube link. So once you have this website, that will help you to find all the tools today we did a training on, and I will continue improving on. And then if you have questions with new additional functionalities, contact me with this email. Also, you can send a request directly submit on the gobiiproject.org for your new like the consulting for tool feature enhancement, always bug reporting. And these presentations on the flash drive in the folder. So you have everything you need. So in this Galaxy, we'll show you the GS Galaxy pipeline. We have some functionalities, including the pre-processing for your phenotype files, that including blue blob calculation or genotyping file you want to do encoding or imputation, and also all the key functionalities for prediction, for model training, for cross-validation, etc. So as I mentioned, there are development server and also testing server, production server. So the previous slide I showed you. Today we'll push the tools onto the production server, and then you guys will be our first group people to pressure test. Let's find out. Okay, yeah. So we have 4 scientific developers located in the 4 of the hubs, including IRRI, Agricide, CIMMYT, and also Cornell. Each of them were responsible for certain tools from their CG or they have the expertise with. So Anhao developed the GBV prediction, blue calculation, also cross-validation, working closely with Kelly Robbins. He's our module file lead, and also working directly with CIMMYT, BSU team, biostatistics unit. So Venice here I already mentioned, so there are plenty genotyping processing tools to develop, also hosting the EIB demo server for supporting more broad users. So Siva worked on imputation tool. The tool we are going to do demo today didn't have this installed yet because still have some bugs to work on. And Victor worked on sample matching to bring your phenotype, genotype together. There's still a little bug we need to work on. So today, the hands-on demo mainly the tools developed by Angel and Venice. So we have 5 meat breeders. They are the GIS implementation breeders and also our alpha testers for this Galaxy pipeline. They work closely based on the steering team from each of the CG in terms of the more higher level the strategy, how you want to implement your GIS based on customized breeding programs needs. And altogether, we fall in under GOBii. So Liz is our director supporting our GS pipeline development, and together we fall in under EIB-5. And as I mentioned, this pipeline really is using a lot of existing tools. So we have a lot of contribution from the expert statisticians, including Pancho, Paulino, so you know BGLR developer, right? So they are the owners of many tools, including the Blue Blob and also the GOBii BGLR. And we welcome more people to contribute. So T3, so Jean-Luc's here, his postdoc participated the testing, and we also have from Cassava Base. Cassava Base has integrated the GS pipeline. We work closely with Cassava Base GS developer as well. And we know there are many functionalities some of you are aware of, so continue give us feedback by submitting on the GOBii project website. Yeah. Any questions before I move on? Okay, if not, so where GOBii Galaxy and IC pipeline start and stop? So currently this is still a stopgap solution. You may already have your data management system, or maybe you don't. Everything's on flat files. So we enabled both. So Galaxy pipeline is just in this corner. If you have the files already, have the format, we will tell you what's the default format, and you can run this as it is, and then you can have the output from the pipeline, and then you can go back to do the selection. If you already use B4R, or BMS or Cassava Base or any kind of breeding data management system, you can go through the, like say for your phenotype, you have the 12 from your field book, and then you may do the extraction based on your 12 design, that whole data set, because you may have multi-year, multi-location when you did a compiled data extraction. So they will come out, you as a breeder, you will know how you want to predict, because not everything you put together will be relevant to each other for you to do the analysis. So as a user, you are still the driver, even though we give you the tool, we give you a car, but you really know how you want to drive, how fast you want to drive, and where you are going, okay? And then yesterday, we showed you the sample tracking. So sample tracking in commercial company is directly in the breeding data management system. So as breeders, you design the trials, and you know what material you want to do genotyping, and then you place those trials in the genotyping experiment. They come through the vendor, right? You receive the data. So if you have GOBii data already loaded into GOBii, extracted in GOBii, and here when we extract, you can extract within a data set. That's how you load it, or across data sets. You may have the germplasm genotype across many different types of platforms, for example, GBS, or just a few hundred SNPs, or maybe SSR, etc. So you can extract based on this list of the germplasm, and you receive a compiled extract file. So from there, we have two extract formats. One is the flapjack format, one is HiMap format. So the flapjack, this morning you already see, basically very simple. You may have the just in the rows are the markers, and in the column shows the sample name. And HiMap, in GOBii HiMap, we have 36 rows metadata on top, and you have 11 columns for marker metadata on the left. And then your marker matrix is the tabular format. So that's the intersect. So when they come out, for the current GS pipeline, we use HiMap or type delimited. So if they are extracted from GOBii, if you do not have these extracted from GOBii, we tell you what's the default input file to run each of the tools. But this is overall our vision. You can still do this GS pipeline as a plugin to enable more streamlined process, or as it is, more like stopgap. And we do have some recommendations for visualization. Today, I may not have time to show you, but one tool we already worked with CINID maize breeders called Tableau. This is very interactive. You can have your output file from your multi-year trial, and also the pipeline, GOBii, all the output put together, and you can select, do your advancement just as how you use your Excel file to look at many different column trees and look at your seed sources, you can go back to your next cycle of the breeding pipeline. So that's where the GOBii Galaxy pipeline start and stop. Any questions? Okay. So if not, just very quickly, we have 3 workflows in the current Galaxy pipeline. So from high level, basically it looks like this. You upload your input files, and then you can select the tools and the parameters. So I will show you the tools in the pipeline. We call them component tools. So then you can choose the model. There are several models, mainly the BGR models, and then you do your prediction, and then you can do your cross-validation while you train your model. Then you export the output file, and then you can export that just as type delimited or Excel file. And then if you do use the visualization tool like Tableau or Flapjack, you can have all your different column phenotype and also predicted values put together to do visualization to select. So this is the high level of the GIS pipeline workflow. To break down this for different scenarios, we start with simplified. The first workflow is you have very well defined training and prediction data sets. That means you are very clear about your germplasm, what you want to train on. So you have the material already been genotyped and phenotyped, and you also have the materials only genotyped. And you know these 2 sets of data sets, the individuals, they are quite related. If you want to use the called the testing data sets or training data sets to predict the data set, you only have genotype and not the phenotype, you know your results is going to be very informative. So this is called a very well defined training and prediction data sets. If that's in that scenario, you use workflow 1. So the workflow 2 is your population is very diverse. You have your germplasm across many different years of breeding programs. You want to know how these lines related to each other. So we have Assignable Tools. These tools are looking at genetic similarity, looking at PCA, and looking at classroom tools to help you group them based on the genotype matrix file. Okay. So you use that information, help you to explore. Then you will say, okay, this is my subset, and this subset will be relevant to each other for me to use workflow 1. All right. So workflow 3 is you combine workflow 1, workflow 2. So once you know the relatedness of my germplasm, you will say, I give them a grouping membership. So this grouping membership can come from your phenotype or based on the pedigree or based on any kind of classification you're aware of. So the example I want to give is like, say, for semen maize, you have yellow, you have white maize. So you can say, I want to predict within yellow, within white. Or you say, I just want to predict all the optimum conditions within each other because not relevant you use your drought condition data to predict your optimum testing condition. The same here, you may have salinity and you may have irrigated or so things like that. So the user-defined training data set can be from your trial design or can be from the population structure analysis based on your marker matrix, based on pedigree. As long as you give a grouping membership, so use that as input, then you can still choose your model, do the training and the prediction. So these are the 3 workflows. So workflow 3 is more complicated, so you can predict based on within or across groups. Yeah, is that clear? All right. So if clear, let's go to next. This is very busy. You do not need to see details, but roughly, okay. So if you use GOBii, you're the GOBii user, right? So you're looking at a GOBii data extractor. You're also looking at a Galaxy GIS data, and then you upload into Galaxy, and then you can choose the component tools, and then you run your pipeline. So right now, we are still not there. What we can do is you have your extracted file saved on your local desktop or the flash drive one I gave to you, and then you upload on Galaxy user interface. So this is run on the server, And then there are different tools, step by step. So you understand what tool need to do what and where they need to come in place. So you are still the driver. You need to know, okay, this is the brake, this is the accelerating, and I need to really turn on the AC. I need to do the wiper. So whatever you need based on the condition, so you will choose these tools. So once you have all the outputs, you say, okay, now I got to almost, okay, that's my final destination. Now I need to really export the data, use a visualization tool, and then from there, I want to compile all my information, then you can make a decision. And then our goal is we do not want every breeder spend several hours just to learn all this. Our goal is we will have this assembled in the sequence as a few default pipeline. For example, the workflow 1, 2, 3 for rice or for maize or for chickpea, cassava, whatever it is. So eventually our testers will assemble a few workflows. These workflows will be like what Milka this morning showed you. You can just get them downloaded and then on your workflow, the one shared with you, you just click a button that will run through the analysis, the type of prediction problem accordingly. So that's our vision. We are not completely there, but we are very close. Today, the tool I'm going to show you 3 major tools. One is for GOBii, one is for blue blob calculation, one is for GOBii, one is for cross-validation. And on top of that, a couple of genotype pre-processing files that basically doing encoding or doing imputation, okay? So I don't want you to feel it's too complicated. Eventually, breeders only maybe have just 1, 2, 3, the 3 workflow you only need to use once you understand how this car function. Okay, that's where we will be. So just a little bit, 2017, our timeline, we had just BGRR wrap, and then we have the K-cross validation. And this year, the items highlighted in green, very exciting. This morning, we were able to push the tools available onto the Galaxy server, so we make this as green. But you can see we still have several functionalities still in active testing and development or testing. So our goal for 2019 is we want to pull data sets directly from the data management system to simplify the file pre-processing. And we also want to improve the Galaxy UI, so make it less complicated. And we like to collaborate with statisticians, including GOBii. And for these output results, we want to enable you to push the result to store in your data management system. So this is roughly our pipeline and our deliverable for the project. Any questions? Yeah, go ahead. Deliverable for the project. Any questions? Yeah.
So is there any difference between GEBV value and GBLOB?
Yeah. Yeah.
Any difference?
So GEBV,
so what's that?
Right. So GBLOB, you need a G matrix to be calculated, to be part of the GBLOB calculation. So currently the GBLOB only use your phenotype and use your marker matrix, not calculating the relationship matrix. Yeah. We will show you. That's why we say our GBLOB is still under development and testing. That's very similar like RKHSS. Yeah. All right, so this is a little bit like a benchmarking or outlook. The one shows yes means you can see for Tyso can do GWAS, and the next gen cassava has the SOL GS, that's the integrated GS pipeline. So currently GOBii is somewhere between the short term and long term. So for the long term, we like to give you more user-friendly type of UI and also capability to handle large files. So other functionality, you can see we are still a little bit behind in the crop-specific workflows that rely on the GIS testers from each of the crop to work with us, so that more readers will be able to use this as a push button type of tool. and also GOBii multi-year environment, we still need to work on. So currently the tool is still very basic. So we make sure it's very simple and very easy. We want to make it easy, but we are still a little bit away from that. But you can see many key functionalities we are already in place. All right. So I think I will stop here now.